Hello everyone, this is Dr. Fox, and let's talk about thrombocytes. In this video, we'll describe the structure and function of thrombocytes, their development and factors affecting their development, as well as the role of thrombocytes in the clotting process. We'll finally discuss the conditions and risks of thrombocytopenia and thrombocytosis. So thrombocytes or platelets, these are two terms that are perfectly synonymous with one another. These are formed elements within the blood that are just little bits of cytoplasm, so remnants of the cells from which they are derived. They are a key part of the hemostasis process or clotting. So if there are any discontinuities in vasculature, it's the thrombocytes that are absolutely vital in plugging these discontinuities or holes. Ultimately, they are derived from a line of cells that give rise to megakaryocytes, and this process begins with hematopoietic stem cells. They function by means of degranulation. So while they are anucleate and they do not possess much of the cellular machinery that other formed elements may, they are capable of releasing substances to encourage the clotting pathway. They don't last a particularly long time. In fact, generally a week to 10 days is all that they will live within the blood before being recycled by the spleen. The spleen is an organ of the lymphatic system. It exists in the left upper quadrant, and it has a major role in the storage and ultimately removal of thrombocytes from the body. The development of thrombocytes is fairly interesting. So we start with the pluripotent hematopoietic stem cell. And then, as you know, there are two pathways. They could either go to the lymphoid stem cells or myeloid stem cells. For platelets, we follow the myeloid pathway. These become megakaryocytes, which differentiate into megakaryocytes. The megakaryocytes then transform into proplatelets, which then shed little bits of themselves as platelets. These cells are hanging out in the periphery of the marrow cavities at the ends and the spongy centers of various bones, and they shed platelets, proplatelets and platelets into the sinusoids. This is a process which is controlled chemically by thrombopoietin, or TPO. So TPO is going to enhance megakaryocyte formation and activates them into forming more proplatelets. Now, within the spleen, there are specialized macrophages which are going to phagocytize old thrombocytes. So they will engulf them, break them down, and we can recycle parts of their cytoplasm ultimately to make more thrombocytes. The clotting pathway can be very complex, but we will simplify it to the best of our ability in the context of an anatomy course. So this all begins with vascular injury. So there is some sort of discontinuity within a blood vessel. Now, what happens then is that platelets can begin to bind to collagen, which has been revealed by this injury through an intermediate, and the intermediate is called von Willebrand factor. Now, as they begin to bind to collagen via von Willebrand factor, their form begins to change. They become a little more sort of dehydrated-like. They develop things called pseudopodia, or false limbs, and they begin to degranulate fibrinogen. And what it does is it's worked on by thrombin to convert fibrinogen, which is soluble within the blood, to an insoluble protein called fibrin. Thrombin converts fibrinogen, which has been degranulated from platelets, into fibrin. So soluble, so dissolved in the plasma of blood, to insoluble. That insoluble fibrin forms a mesh, and it really anchors more thrombocytes into place, which is the basis behind coagulation, that is the clot. Now, thrombocytes are more abundant than leukocytes. They are less abundant than erythrocytes. So they come in at about 150 to 400,000 thrombocytes per microliter of blood. Any less than 150,000 thrombocytes per microliter, and that's a condition known as thrombocytopenia. So thrombocyte is the platelet, and openia means a shortage. So people who are thrombocytopenic are at a greater risk of bleeding and or hemorrhage. The opposite condition is thrombocytosis. So that root there, cytosis, means an elevated level of thrombocytes. So this is when there is greater than 400,000 thrombocytes per microliter. And there are two possible outcomes with this. One is spontaneous clotting. Spontaneous clotting is very bad because clots that form within vessels can then move to other areas of the body where they can cause a problem. So one such example of this is a deep vein thrombosis of the lower limb, which can then travel up to through the heart and out through the pulmonary vessels out to the lungs, where they can become pulmonary embolisms, which are life-threatening. The other possibility with thrombocytosis, and this is rather interesting, is something called paradoxical bleeding. With paradoxical bleeding, the clotting pathway becomes dysfunctional. And what actually happens is all of the thrombocytes begin to overwhelm von Willebrand factor, and there's no room for von Willebrand factor to bind to collagen. So it can't act as an intermediary between the vascular injury and the thrombocyte, and as a result, the wound, the discontinuity, never forms a clot and never heals, and as a result, there can be bleeding. So let's evaluate some of the information that we just discussed. With which blood protein must thrombocytes interact to form a clot? Albumin. Albumin is an important blood protein, but it is more important for oncotic pressure. Fibrin, yes, absolutely. That is going to be an important part of the clotting pathway that forms that mesh network into which thrombocytes are captured to form the clot. And then the other two are just antibodies, IgG and IgM. No, those aren't necessary at all. So the correct answer is fibrin. Thank you so much for your time, and I look forward to talking with you in another video playlist. Hope you all have a wonderful day.